right, well, thank you all for being here. We are really excited to hear that you'll be attending some patient conferences this summer, whether it's your first time or you've done this many times before. We hope that you'll leave this orientation with some new insight that maybe you didn't have before. So my name is Katina Kalakos, and I'm here with my colleague Misha Kwanda as well. We're both scientists on the Searchlight team. And again, today we'll be presenting a little bit on recommendations and orienting to participating and presenting in patient conferences this season. We also have Julia Summer here online from the Simons Foundation, who is very collaborative with us for all things family meetings and biorepository, and also is better suited to answer any questions that might come up pertaining to SFARI and SFARI grants. So thanks all for being here, and we'll get started. So overall, our goal is to review the landscape of patient conferences, chat about engaging with families throughout the conference, and then the last part will be presenting your science to families affected by variants in the genes you study. We do know that not everybody on the call is necessarily presenting science to families while you attend, but whether you are or not, we hope that you'll find this helpful just to consider how families are receiving science when they attend these conferences. And so to start with landscape of patient conferences, it really is quite variable what the conference will look like. And particularly conference size and the activities that happen at these meetings, it really varies widely based on the organizing group, typically a patient advocacy group, although they might be collaborating as well with an academic institution or other institution. It depends on the number of families that are engaged both in the patient advocacy group network and just attending the conference itself, as well as the number of researchers who are engaged in the space. And then the amount of funding is a big predictor of what the conference will look like. And so considering these factors, conference agendas usually have one or more of the following features. So at a minimum, and this is a really central, critical part of the conference, there will be family-only meetups and fun family socializing. As you may appreciate, the families that attend may never really meet another family affected by the same condition as them, locally, even in their state, maybe even in their country. And so this is a really great opportunity for families to connect, for the kids to meet each other, and really build that network, both kind of locally, if that applies, and internationally. It's also just a ton of fun. If you haven't been to one before, you'll see the kids are running around. It's really a good time. On top of that, what you'll see, given that US scientists are attending these, you'll likely see some speaker sessions. So this is where the researchers are presenting their science to families, oftentimes in a more lay-friendly way than they would when speaking with other scientists, although there's a bit variability that we've seen at different meetings. There are also sometimes scientific roundtables. And so this is where the researchers are presenting science to each other. So we can dig more into the weeds of the technical terms, talk maybe more about preliminary published data, form collaborations, kind of workshop next steps for future scientific directions. And usually if this is happening, it might be the day before all the families come, or it might be on a day where the families have some other like fun activity, maybe like a museum outing or something like that. And then sometimes you may also see some on-site activities related to research and clinical care. So there might be some on-site research data collection. We've seen all kinds of things. We've seen mobile EEGs, neuropsych evals, motor testing, blood draws for the purpose of subsequent biospecimen development. There's also sometimes on-site workshops related more to clinicians and therapy. So we've seen physical therapy, OT, speech therapy workshops that sometimes experts will provide to families, or sometimes families make their own workshops and present it to each other for kind of common skill sharing and skills building. And then we've also seen some clinical evaluations happen as well, most often in a research context, but sometimes outside of a research context. And so to dig in a little bit to kind of how families are receiving researchers at conferences, we will say, and I should have said this at the top, that much of what we're covering today is based not just on our own experience working with families at conferences, but also we've like explicitly solicited feedback for the purpose of this presentation. So much of this is coming direct from families, and we're just serving as a conduit of that insight. Um, Researchers are entirely welcome at meetings. It is really meaningful for families to know that people are invested in understanding a gene that affects their family. And so while we understand that you might be busy, you might be flying in, flying out for a brief moment, if you have the time and if you're comfortable, we really encourage that you stay beyond the presentations. Consider sitting and talking with families during meals. Lunchtime is great. There's also frequently childcare happening simultaneous to the session. So sometimes parents or caregivers will have more opportunity to connect more directly. And also, it's a good time to talk to other researchers, right? Especially if they don't have if the conference does not have kind of a standalone scientific symposium. We also want to mention that it's really important to include the affected individuals with the genetic condition in the conversation. It can really be isolating and dehumanizing to avoid conversation or eye contact with people with neurodevelopmental disorders, especially if those folks are adults that are attending the conference. They might be with their parents or they might be by themselves. But just keep this in mind to include the affected person in the conversation. It could be something simple as saying, hello, what's your name? High five. Um, so just to be mindful of that is really important feedback that we got from parents. And so for additional considerations for engaging with parents and patients at the conference, again, solicited from feedback from parents, as I've said, many families are just super jazzed to talk to researchers, like truly excited that you're there. They really want to learn more. I mean, they really want you to learn more about their children and how living with the variant in the gene that you study affects their lives. So outside of just the laboratory context, but really seeing in person what that looks like clinically. With that in mind, we've heard that it's really important to not assume what the top concerns or needs are for families. So, for example, some conditions might involve epilepsy, but if the epilepsies are pretty well controlled, then the families and that group might actually prefer investing studies in improving communication, right? So, just important to connect with families and connect with the patient leaders in the space to really understand what's most important to them. And one way of doing that is simply just asking open-ended research-related questions, and hopefully that can help answer some of those questions. So a few examples we have here are, what is it like for you to live with this condition? Or what is it like for your child to live with this condition? What's been the hardest thing about your experience? Is your experience shared with others or is it unique in some way? And if so, how? So, just some examples here is maybe that particular family is affected more or less severely than the average person that we have in the literature with the condition. Or maybe there's multiple people in the family with the same condition and maybe it manifests similarly, maybe it doesn't. So things like that. And really, this last one is important. What do you think is the most important thing researchers should focus on? This is a pretty simple and straightforward question, but it really unlocks a lot of important insight that might be valuable for forming future scientific and clinical directions. Now, in terms of asking questions, in general, we've heard that no questions are off limits. Um, I mean, outside of like the, average social norms of asking people questions, parents want to answer your questions so that more information is known by researchers. That being said, uh, families may become emotional. We see this happen. There's a lot of excitement and there's also often a lot of tears at these meetings. It doesn't mean anything happened. It doesn't mean anything went wrong. It really just means that it's important and it affects them. So our recommendation from the parents we spoke to is that it's okay to, allow a bit of silence, a little bit of quiet, just to have the person gather themselves, right? It's important to express empathy, not as much sympathy, which we can get into a little in the next slide. And if it feels right, continue to talk to them, right? You don't have to fix it. And parents really don't expect you to fix it. They just want you to listen to them, and they want to have a conversation that's meaningful for both of you. We asked a little bit about what parents have found to be helpful statements versus maybe unhelpful statements. And so we have a few examples here. Some helpful example phrases have been, I can see that this has been a really hard journey, or that sounds challenging, or you've been through a lot with this process, right? Expressing empathy and kind of understanding their experience and centering that. What we've also asked was about some unhelpful phrases, and the families know that this is always with good intention, right? And we all know that this is usually with good intention. It just doesn't always land well. So a few examples here are leading a sentence by saying at least. So at least your kid doesn't have seizures, or at least they're able to walk without assistance, right? Like, so, it can minimize that person's feeling and experience and sort of contrast that in a way with other families that just makes things uncomfortable. Um, Offers of unsolicited advice or recommendations, especially if not a clinical expert in the area, was shared with us as unhelpful. And getting back to a point from earlier, statements of sympathy, which again, we recognize are well intended, but sometimes land in a way that can convey pity or can make families feel distant or distanced from you. Their experience is not as relatable. So things like, I'm so sorry, I cannot imagine what you're going through, right? Like it's not within the realm of possibility for me, or I feel so sorry for you. So this is what families shared with us were not helpful, even if well-intended. Similarly, we asked about, what else might you want researchers to avoid? Um, we have found that sometimes families respond poorly to very pathology focused language, even if it's scientifically accurate, right? We understand that many of the terms we're using here are very classically used in biology. They just don't land the same when talking to a room of families. So a few examples here: genetic mutations often does not land well with families. It can be off-putting. People think mutant, they think alien. Again, we're thinking more popular culture and public consumption. So we recommend using phrases like genetic changes or variants, and avoiding the word mutation. Terms like dysmorphic and misshapen, again, if in a context of a particular diagnosis or evaluation, that might make sense, but it does sound a little harmful to families, so might consider some more neutral language associated there. And then terms that, again, might be rooted very clearly in biological process, but don't sound great to families. Terms like lethality or early cell death. If needed to use to properly convey the science, we understand, but then really define it very clearly and in lay-friendly terms in order to mitigate concern, because it can happen really quickly that families misinterpret something, and we've seen the atmosphere really change quite a bit, whether it's on a one-on-one interaction or during a scientific talk. So again, all of this is really just to ensure that the families feel okay and that your science is being received well, since you are there and taking the time to be there. The parents also have recommended that we avoid statements outside of the scope of research. And just, when talking about research, just consider the potential impact of it on the families, right? If there's certain elements that when reviewing and thinking of the lens of a parent, if something sounds like it might be harmful or might be misconstrued, either avoid or consider how to modify the language. And relatedly, the family shared that giving out advice outside of scope of expertise is often not helpful. So we recommend perhaps providing that level of advice only really if families ask for it, and also only if you're comfortable, right? Definitely okay to say not my zone. Um, and so, much of this we're talking about, but really just to reiterate, words do matter. Scientific language can be interpreted differently by families, and in the worst case scenario, can alarm them significantly. and perhaps in ways that are truly not warranted and not intended. So we recommend being specific with word choice to minimize that risk of causing families undue alarm, especially. So two more examples we have here, using the term neurodegeneration. Again, rooted in biological process, rooted in clinical expertise. Um, in some cases, right, neurodegeneration does properly reflect kind of what we observe in a genetic community, and sometimes it does not, but it might be used to, to talk about a specific process. So just, families just remember that families might think that the whole condition is neurodegenerative. And so if it does not apply there, we just recommend not using the term or thinking of other ways to define it more clearly. One more example here, we've heard, this gene is involved in a cancer pathway. That may very well be true. And that may really inform and direct the kind of science you're doing. And so it's important to understand that. At the same time, families might think when they hear that their children will develop cancer, right? Just again, thinking of ways in which it can be kind of interpreted or extrapolated beyond what you might have meant. And so it's they might even think that even if there is no clinical evidence showing elevated cancer diagnoses to date. So, consider words and then consider additional phrasing or additional explanation if something like this is important in your scientific talk or in just in your conversation with families one-on-one. And one last slide from me before we pivot into presenting science to families. Uh, you know, Often families with autism with a genetic etiology will have a different perspective than folks with idiopathic autism. And so, what we have found is that families with profound and more disabling autism may sometimes find the neurodiversity movement a bit off putting or a bit unrecognizing of the life changing impact of autism in their family. That is not to devalue the work of neurodiversity advocacy. It's not an either or, right? Both are important to uplift, but it's important to understand the audience that we're talking to in a particular conference or a particular interaction. So in this case, you know, given the conferences you're likely to attend, we do recommend and families recommend avoiding too much centering of neurodiversity advocacy beyond that which is representative for that particular community of interest, right? Even if kind of more globally in thinking of autism research, this is, an off talk about a topic, an important initiative to continue. And finally, again, depending on your comfort level, you don't have to do this. You might consider bringing business cards to share with families that want to follow up with questions afterwards, only because the families, again, are really jazzed by the science. Families are becoming more and more fluent in scientific language and reading papers. And so they remain intrigued and interested to hear science updates. So if you'd like, you can do that. If not, also fine. So with that, I think I'll pass it over to Misha to talk about presenting science at patient conferences.
Yes, and thank you so much, Katina. A lot of what I'm going to cover kind of melds into what Katina introduced previously. So I'm going to start with very general considerations. So the general consideration and the most obvious one is this know your audience. And I mean that and I mean that in the sense of, are there going to be parents in the room in addition to, are we presenting to just scientists? So of course there's the science roundtables where there might not be any parents in the room, but there might also be these scientific conferences where it is presented to other researchers, but families are welcome to attend. In addition to the parent only or the family only presentations. So when families are in the room, just like Katina was saying, the way the data is described, not to entirely take away from the scientific jargon and the way it's presented, but in particular about how the affected person is discussed. You might want to consider that a little bit more when they're in the room. Words like proband or patient kind of distances the research away from the loved one or the person and the reason why they're there to learn in the first place. Another general consideration for presenting now for more of a family audience, or when families are in the room as a primary audience member, is having sort of questions throughout the presentation for engagement. This was feedback again from the families. So pausing and saying, what did we learn from this experiment? Or why are we doing all this testing on mice? To recenter sort of what the point of what you're trying to make is. And I'll come back to this a few times in my section, but in a 2-hour, 3-hour, 4-hour day for these families, when there's all this new information that's coming, sometimes finding ways throughout the presentation to bring back to the main focus or highlight the overall point of what you're trying to overall describe can be really helpful for this lay audience to engage and re-engage if they've drifted off or missed something. And then finally, the obvious, leave enough time to field questions at the very end, because the families definitely will have questions usually at the very end. So now when it comes to knowing your audience, so we recommend, and our IRB recommends, this 7th grade reading level max. And that's not to say that it's dumbing down or that families can't handle a higher reading level for educational purposes. It's more that you're creating a neutral level away from this sort of specialized space. So now that this is such an international community, as many of our rare communities are, there's many families that are not native English speakers that are coming to all the conferences. And so they might not have been educated in science, let alone in English. So it's much more helpful to these families if the presentation is at a lower level. In addition to, it doesn't mean that they're uneducated. If I went to a law conference or an environmental conference, there would for sure be specific jargon, shorthand that I would be completely lost. And so it really is that science is its own language, and translating that for this neutral environment is the goal when speaking to families and knowing the audience. Coming back to this point that families are really eager to understand the research and how this affects them. And this might be the first time that an expert or clinician that understands the space is speaking to them. Not just the fact that they might have been diagnosed last week, last month, because we see that at conferences all the time, that someone was diagnosed last month and all of a sudden they're attending this conference. But just that locally, they likely don't have an expert in their space that is able to take care for them. So they really are, if they're showing up and they're coming, they really want to understand the research that's affecting them and their families. In addition to the time of day of your presentation is another know your audience sort of tip. Earlier in the day, you likely want to do more background, in general, like a genetics 101, as we kind of call it, or more background on the condition. Whereas later in the day, you might want to just consider more just your session that you're in with the other speakers, and the general recommendation of maybe connecting with the speakers in advance or getting information back from the conference organizers. So this is something that I have done where I've sent my presentation to the conference organizers and I've said, am I using the right jargon that is being used in this community? Are there questions that have been coming up in this community more and more that I'm not covering or that I should cover? And making sure that I'm orienting it for that community and what that presentation makes sense in the time of day is a key consideration. So now coming to the presentation. So what is the point you're trying to make is sort of the question you want to ask yourself every time that you're putting together a new slide or reevaluating your slides. It's the first 2 points that are two sides of the same coin. So only include things that you need on your slide to make your point. But if you have something on your slide, it's really important that you discuss it. And so I'll have 3 examples that I'll dive into a bit more later. But really having images or pieces of information on a slide that you're like, we won't discuss this, maybe consider removing that or simplifying the slide more so that it's more accessible. In addition to this scientific jargon. So families throughout this long day, through this 4-hour, 5-hour day, is keeping track of their gene acronym, their variant, their protein if it has a different name. And now we've got different researchers in different spaces using different acronyms and jargons. And so just one example, even for me, if I'm going to a conference, ASD obviously means autism spectrum disorder, but it also can mean atrial septal defect. PDA might mean one thing in a cardiac, sort of conference, and to a lay audience, it means something else. So explaining the jargon, but also considering how it's being used and is it necessary for your presentation? Because for the families that are taking in a lot of information, that is a lot more for them to keep track of. And so in this feedback from families, we asked what do families want to know from the presentation? So these are particular points that our stakeholders that gave feedback for this presentation made that they wanted us to highlight, but none of these will likely be very surprising to any of the researchers in the room. The Simon Searchlight community, being the specific community that it is hungry for treatments and cures. That's broad strokes in general, the Simon Searchlight community, and recognizing that other genetic communities or condition communities don't have that same perspectives. But our families in general, they want to know how things are applicable to this goal, this ultimate goal of a treatment or a cure. If you are not an expert in that particular condition and you're coming as an invited speaker as an adjacent pathway or related protein, they want to know, will this treatment work for their condition, either the conditions in the room or the main one that's being focused on, or why hasn't it been tried? Why wouldn't it work? And those types of practical questions, that's what they're wondering. As a basic scientist, and I was a basic scientist myself, they want to know, how does the research answer important questions for humans and for the genetic condition? So as a fruit fly researcher, you're used to seeing all the fruit fly researchers present why fruit flies are a great model organism for human disease modeling. But not just that, they want to know, the families want to know more, like, why are you testing protein levels? Why are we using cell models? Why are we doing animal behavior, and how will that ultimately help them and their child for this treatment and cures goal? Some of these connections might seem really obvious to us as researchers, how this is important in the bigger toolbox of what's needed to be understood and known. But in a sort of basic science dense presentation, it's really not often obvious to the non-scientific families in the room. And then addition to this is making the connecting of the dots. So what information is still needed to be known for this next step, for this therapy, for what has to be tested in animals, and then it's tested in humans, and how does that work in order to get to that ultimate goal? And so now we're also approaching the perspective. So when a scientist goes to a scientific conference, we're used to presenting to a room full of scientists, and they're skeptics in general, right? You're defending your work, you're trying to sell the value. You definitely over-explain, or not over-explain, in the scientific realm, how you got to that point and how the methods and data were created. For families, it's sometimes more information than they need in terms of methods, focusing on the data and the results are more what they want to know, but they're coming in with hope. So the perspective and balance when presenting to a parent audience or when parents are in the room sometimes is a needed consideration, because these families are not coming in as skeptics. They are the general theme of hope is a common one at a lot of Simon Searchlight related conferences. So making sure to not overpromise and ensure potential benefits are grounded in this reality, as well as not going too much into the nitty-gritty to make sure that you show the value and the importance of a particular research method. And so I think this is my last slide before going into some of these examples. But the last consideration is, what point really are you trying to make? And sometimes it can be really useful to have a non-scientist look over your slides, give you some feedback. Consider take-home points, and I've seen this done really well at many conferences recently, where researchers, they give their presentation with all the data, and at the very end, here's what we want you to walk away from and know from this presentation. Because again, this can be a lot of information for families to take in, and they might be worrying that they've missed something. So the overall take-home points can be really helpful. The implications of research, as mentioned, rather than the methods, can be really important and a key sort of focus. And then finally, this analogy or comparison. So I'll dive into one of these examples, but when there is a key experimental or methodological or function of the protein that needs to be known for a gene or protein or a pathway that families really need to understand in order for you to show your data and make your point, finding and getting creative with an analogy or a comparison can be really helpful in their understanding. And I'll give one example in my second example coming up. So what I've done for the three examples is I've just taken figure papers. So these obviously are not presentation ready. Likely the researchers are would present this differently if they were presenting to a family audience. And they're research papers that have used Simon Searchlight data in the last few years. So as you can see, not surprisingly so, it has a lot of data panels. It relies on assumed knowledge of just in general how to read a graph, like x-axis, y-axis, experimental methods. The title is the figure title, so it has scientific jargon, and it has minimal explanations in the text on the slide. So if you go to how we would modify this, so if we were jumping in, and again, families came in and made some edits and helped us with this, but we would obviously want the title to be the overall point of the slide. You want to adapt your figure, just show what is needed for the point, for the most important piece of data for the take-home message. Relabeling the X and the Y axis so that it is more readable and understandable without having to know the acronyms for that experiment. And then we also suggest spelling out a little bit of information on the slide about what the point is. And part of the reason is not to read it out and make sure that it's word for word, but what we find sometimes is that families will be taking pictures of the slides, and I think you likely have seen these at these conferences, and some are note takers and others take pictures. And part of that is they're trying to make sure that they're getting all the information and they're understanding all this information, and having a little bit of text to orient them to have a little bit of background can be really helpful for them in the future when they're trying to refer back or go back to make sure that they've understood the main messaging of that data point. So for my second example, now taking from a different paper, channelopathy, it obviously has a lot of data panels, scientific title, assumed knowledge of electrophysiology, and unexplained acronyms as before. And so now if we were to modify this, and I've divided it into two slides for my example. So giving a little bit of background with this, type of analogy that is a little more neutral, that can be accessible to the general population as to what this sodium ion channel does, what it looks like when it's usually controlled, what it looks like, what it might be functionally doing when there's a loss of function, gain of function, and explaining that as a background that then slide 2, Katina, next slide, that then can be carried forward to explain experimental findings. So then relating this back to what does it mean when you show a wild type or a general population? It's the usually controlled channel function, whereas this other variant results in a channel that's open longer. So you have more of a gain of function effect. And relating that analogy as you go through your presentation can be really helpful, so that it's not just, oh, they got the concept and we're moving forward with the basic science experiments, but you have that analogy and now you can refer back to it as you're explaining what the data is showing. And then for my last example, another common sort of experiment that's seen in the field, just a Western blot, but this is meant to show that in describing an experiment and making something lay friendly, sometimes more words are necessary. So you'll see that in my next slide. But here would be an example in a scientific presentation. You would say that there was less of this phospho-AKT protein as seen by the Western blot in the idiopathic autism versus the sibling without autism. Whereas if you go to the lay-friendly version, obviously we're trimming down, we're relabeling the loading wells, and so that it's as accessible as possible for what's needed to be known. And I'm not going to read out this explainer, but this was written out by a family saying this would be as much of the background that would be needed to help us understand this piece of information. And so it kind of walks through what, how there are proteins, which may have been explained earlier in the conference, but just a consideration about how we talk about proteins, what this AKT protein is, why, what does it mean if it's phosphorylated or not. And so when making something lay friendly, it is more work because you can't use that shorthand scientific jargon that we all know what it means, and we've done years of experience, years of education to try to attain and come to that level. But for the families, you have to use a lot more terminology and explainer to get them to that point, to get them to understand the main data point for a presentation. And then for my last final slide, just some after presentation suggestions, just like Katina said, there will be families that will likely walk up to the podium and speak into the microphone and ask their questions. There will also be many families that are not as comfortable for a multitude of reasons, but they would like to talk to you at lunch or informally. So being available afterwards could be really important and not rushing off too quickly. But this other idea that kind of came out from the families, which is this openness for collaboration. And they don't mean openness for collaboration in the same way of we want to be on your research papers. They mean that they want to have an exchange of ideas that can be useful for their genetic community. So some ways that this might look is that they're coming forward and saying, often in our community, this is a clinical feature that's being reported, but we're not seeing it in research papers. Have you seen this? Could you look into it? Is there data here to investigate? That, or in this example that's highlighted, this Coulind-de Vries syndrome researcher started a retrospective MRI analysis because they'd communicated with the families and they learned that families actually do have their clinical MRIs. And so this researcher was able to start the research study because they were able to get this information from the families. So it's really, and coming back to more of this, what questions are interesting to the families, or what questions are the families wanting to be answered, and not necessarily this, where is the science leading us in curiosity. And so that's what they mean when they say openness to this explore collaborations. Really this exchange of ideas and working in partnership to make a difference for their loved ones.